Huang. I'm the assistant professor of biological engineering at Utah State University, and today I'm going to introduce the recent work from my lab and establish an efficient cancer invasion model and study the chemotactic migration of glioblastoma, also called GBM. So GBM is one of the most prevalent and aggressive brain tumor. And as you can see, unlike other tumors, these GBM tumors has no memory boundary. So that means is, the individual's cancer cells can actually migrate, invade into normal tissues, and be able to generate new tumors. That's why eradicate these kind of tumor are extremely difficult because individual cells can actually invade and reoccur and into individual tumors. And then this is one example. You can see in the scalp, the GBM tumors can invade into the skin and form new tumors, and extremely aggressive process. And if you're looking at the histology of these tumors, you can see they actually invade through individuals' cancer cells and be able to infiltrate into the normal tissues. That's why they're very aggressive and very hard to remove, right? And in 2013, people noticed their individual infiltration process is very unique. As you can see, the cell morphology is quite different from what you usually see for other cancer cells. And what happened is they can actually squeeze the cell body into the interstitial cell spaces and be able to drastically change the volume to adapt the migration paths. So almost 10 years ago, we started this project by designing microfluidic devices which can use PDMS microchannel to mimic the interstitial spaces for individual cells to migrate. So this is what the cell device will look like. So you have cell culture from one side, and see the cell can see it from one end, and the other end has no cell to represent the normal tissues. And so between these two channels is connected with micro channels, which is smaller than individual cells, function as interstitial spaces. And for the cell, in order for the cells to migrate over from the seeding side to the receiving side, which is normal, representing the normal tissues, and the cell has to drastically squeeze themselves into those microchannels and drastically change their volume. All right. So this is what we see. We can monitor individual cell migration process through the microscope imaging, time-lapse imaging. And as you can see, you start from the top left corner, and then individual cell be able to stretch themselves into these really small spaces and migrate in a well shrinked manner and through the other side. So using the system, we can monitor the whole process and trace individual cells and understand the migration speed as well as some other biophysical properties of the cells. Now, lately we start thinking, this process is automatic process, so spontaneous process does not involve any chemotactic factor, which is not exactly replicating, what happens in vivo. So we're wondering, can we implement, chemotactic agent into a system to be able to also replicating the biochemical process, right? Here's what we do. We're still taking these devices, and instead of having enclosed chip system, we also have, the medium with different component to slowly perfuse this channel. So make sure those channel dimensions are constantly replenished with the conditions we want. All right. So we use fluorescent molecules to model this process. This is the to represent how the protein would diffuse into the system. Right, and as you can see, over time they can generate. If you add zero concentration on one end and 100% concentration on the other end. Over time, they can perfuse through this channel, through the diffusion process. At the end, you can see it forms some sort of a gradient, right? And through the image analysis, you can see this process, again, they're representing, the fluorescent intensity of the molecules. You can see over time, it will form almost a linear gradient. And what that means, if we can control the boundary condition at one concentration, one end, at the other concentration, at the other end, you can form pretty much a linear gradient, any kind of linear gradient. Okay, here's what we're showing. Different gradient can form. For example, if you start with 0, 0, and 100, 100, and that just become, no gradient situation, but has a different concentration of chemicals, right? And on the other hand, you can also form, 0, 100, 100, 200 is the same, which has the same gradient slope, but at a different starting concentration. You can also increase the concentration slope from 0 to 200 and have a heavy and more intensified gradient. So, and then of course, the chemical could have a different diffusion rate based on their molecular weight. So we also did a study on different size of molecules, and we can find out this is showing over time they will reach to linear gradient formation, right? So what we find out for most of protein that involved in the chemotaxis process, you can if you can set up the system for more than 25 minutes, about 30 minutes, you should be able to establish more than 99% of linearity of this gradient. So this is basically showing we have pretty easy manipulative process to mimic the chemotactic process and to generate any kind of a linear gradient we want. So this is what happens once we generate a gradient, okay? And of course, it's like a very typical chemotactic behavior, all right? First, it would respond mostly to the gradient, not the concentration. For example, here is the video we can show as in a different slope of gradient, you can see how individual cell can migrate over. It's very hard to see, so that's why we use the red color to show you where the location are at the moment. So you can see in this race, side-by-side race, you can see, with higher slope, it migrates much faster, and when there's no gradient, it's migrate really slow. Right. So we take the data and to show the relationship as, the concentration gradient and that's the migration velocity in function of concentration gradient versus the initial concentration. As you can see, and pretty much show a good relationship, proportional relationship of these migration to the gradient, but not to the initial concentration. So what does that suggest is, it doesn't matter, what the actual absolute concentrations are. The cell migration are mostly determined by the gradient slope. Another typical chemotactic behavior that we see is, how these gradient can actually polarize the local distribution of the receptors. Okay, here we use growth factor receptor beta FGF. And you can see, and looking at their cell distribution, and they pretty much follows where the higher concentration of the growth factor are. Okay, and we take the data and we tease out the intensity profile. As you can see, for the gradient, you can see the green color, and pretty much the receptor are highly concentrated to the forward direction, and backward gradient is highly concentrated on the backward location, the real location. In the non-gradient situation, it's pretty much a V-shaped balanced situation. And further analysis, you can see, it shows a very good trend that the geometric arrangement of those proteins are highly polarized by different gradient. So next thing we see, okay, now we are able to establish the chemotactic gradient. Can we use that to enhance the capability of these invasion models. Okay, so one of the challenge with the GBM model is that it does not very well differentiate between different different cell lines. Even though different cell lines, they have different capability to actually to invade into normal tissues. For example, here we have cell line 12.01 to cell line 99. So this number representing how aggressive they are in terms of in the patient outcomes as well as the animal studies. All right. So here we show what the typical very classic migration model using the wound healing assay. As you can see, they're very difficult to distinguish between different cell lines, even though the aggressiveness of those cell lines are quite different in the patient. Right. So how can we use the model to enhance that distinguishment? Yes, we can do. I mean, 10 years ago, we started showing in the previous non-chemotactic model, we show simply using the three-dimensional invasion model, we could separate a little bit better in terms of some of the cell line. For example, 33 and 44, we can distinguish that well from 12.01 and 22, but it's very hard to distinguish between 12.01 22, between 12.01 and 22, right? It's also hard to distinguish some other cell lines in a highly aggressive species. So we're thinking, can we by introducing the chemotactic gradient, can we further increase the capacity, the distinction capability of this model, right? The answer is yes. So here's we show how that would work with different cell line. We have 99, 33, and 12.01 as those numbers indicates these are definitely higher aggressive and this is low aggressive. Again, it's hard to see where the cell grows, so that's why we use these arrows to show the process. Hold on a second. As you can see how 99 is definitely moving much faster than the other cell lines, okay? We have done that for many other cell lines as well. As you can see, the results show up here. Pretty much every cell line between any of the two cell lines can be well distinguished using this implementation of the Chemotaxis process. So, and that shows pretty exciting, right? So next thing we're trying to figure out is, can we use that to screen drugs, right? So we haven't quite get to that point yet, but we use resveratrol, which is a very mild anti-migratory compound, and to see if the system would work, all right? So what we're showing here is basically using the chemotactic process at the middle point when it migrates in the center, and we start adding the resveratrol compound and see how fast they actually go back, or how fast the compound can actually repulse the process. Let me just show the video again. Now they start from left end. At the end, in the middle part, they pretty much migrate in the same speed. But once we add resveratrols, they can be repulsed back to the starting point at a different speed. Okay, and in the higher back gradient of the resveratrol, you can see it migrates, the repulsion process is much more effective, right? And whereas there's even though the same kind of concentration, but the different no gradient situation, and you can see their repulsion is very weak, right? Now we also look at how these kind of anti-migratory behavior can how different cell line will respond to resveratrol in terms of the anti-migration activities. All right, and you can see now this is the 0 point is where this is the control where there's no resveratrol treatment, right? And we also treat it with 2.5 gradient slope, which is pretty small, and you can see decrease of their migration towards the positive direction. And then when you add a 50, which is a pretty high concentration gradient slope, and you can see that basically reverse the migration process. of course, you're not going to be able to use the really high concentration, but what really matters to us is, can this compound really inhibit migration in a low concentration profile, right? And we find out it's not, it works in some cases, for example, cell line 22, but not in other cell lines. So this basically providing very good platform to screen this compound, not just the compound itself, but also how show, the patient-specific responsivity. And furthermore, we're looking at, how, what happened with this compound, what things are being inhibited, right? And say a couple things, if you remember in the previous slides that we show, the receptor can be polarized. The geometric distribution of the receptor can be polarized by the growth factor gradient, right? And so, and then we add, on top of that, we add the resveratrol gradient, and hopefully can we see any changes. The result is it basically is still seeing positive polarizations, just like how you see in the positive gradient, okay? And that means, the resveratrol is not directly inhibited receptors, okay? Instead, it's inhibiting more other tumorous genes. So we find out, Ki67, ATX, and PYK2, those are either responsible for the proliferation or the aggressiveness of the cell lines can be detected that's highly downregulated by using the resveratrol gradient. Okay, so in summary, we demonstrate, the GBM chemotaxis invasion models that can mimic, not just the physical microenvironment, but also chemical microenvironment. And it's pretty rapid assay. Now, the video we show you is typically 18 hours long, comparing to the animal studies or other in vitro study that use days or weeks, and we can use that 3 to 18 hours to be able to identify their migration invasiveness, right? And we also be able to well distinguish between different patient cell lines, and that also add potential to future personalized assay. And so we can screen how different patients would respond to different drugs. Okay, I would like to thank my team, and this is the my lab, and then so most of the study has been done here in this project is by Dr. Krish, and he's a postdoc in my lab, and then Dr. Jinan Gen, and Chen Chen is my student in the lab. So Chen Chen and Gen are working on the cell culture and the molecular biology side of the project. While the Krish is working on the microfluidic device design and fabrication process. And also I would like to thank my collaborator, which is a very supportive project, and Dr. John Cole, and he recently moved from UT Austin to China Medical University, and his lab has been very good support to our project and by providing a lot of patient-derived cell lines and samples. All right, thank you so much. Any questions? Yeah.
So for the results, based on my calculations, and they actually reflect the patient, like the middle situation between different patients, the aggressiveness.
Right. This is a very good question. So this is one of the important advantages we find out chemotaxis can enhance this model is that here's, as you can see, in a wound healing, traditional wound healing assay, it's really difficult to show, right? And in most cases, those numbers basically representing how aggressive these cell lines are in the patient outcome, as well as rodent animal studies. Okay, and so you would expect to see a significant difference between these cell lines, and also show the relationship of from low to high respecting to different cell lines, right? But this is not the case here. Most of the cells are difficult to distinguish between them, right? And once we add physical confinement into the model, and you can see it separates a little bit better, but again, they still have a very poor performance on separating 12.1 and 22, which is the low aggressive ones, right? And which is reasonable because they're not stimulated by the growth factor yet. Once we introduce the growth factor gradient, and the stimulation can actually help to separate even the lower migrative species. So most of the cases, 12.1 to 44, at least in these four, you can show very consistent migration behavior as compared to the animal study as well as the patient outcomes. And it's 99. There's a little bit of controversy on the 99, and I think we need to look closely into the animal study in terms of that.
Distinguish, like you already showed that you can distinguish between the velocity, right? Like with the chemotaxis invasion, like the difference between the velocity is very clear. So like, what is the basic application like from this data?
Right. So the question is, we show pretty good distinguishment between cell lines. We can also show very good separation, of cell lines simply looking at their migration speed, right? So yeah, this is a good answer. So, and what we really want to do is, a couple of things. Personalize the medicine's drug screening. So one issue is, people, there's a variety of different cell lines. I mean, we'll be able to get 5 different cell lines, but there are dozens of them outside, right? And from patient to patient, their response to drugs could be quite dramatically different. So how do we really accommodate that, patient-specific differences is, through these kind of personalized screening process. For example, if I take in these kind of patient and into the high throughput screening system, right, we can screen like hundreds or thousands compound. Can we find out the ones that best suit to that specific patient? And I think that's an important question to answer. Okay, thank you very much.